Alright, and welcome back to pain, or I guess technically welcome back to touch. This video we're going to be talking about pain. Alright, so we talked a little bit about pain in the first video. Pain is essentially weakness leaving the body, right? That's what those army posters tell you, right? What pain actually is the detection of potential tissue damage. So this part of the body could be damaged in some way, and we need to look out. We need to be wary of it. So we talked about how there are two different kinds of pain, but I didn't really specify what those two types of pain were, or what those two types of receptors. And I asked you to think about what they might be. So here's the answer. There are two different kinds. There's the A delta fibers and the C fibers. The A delta fibers are basically the pain that you experience immediately. So, for example, if I were to thump you right in the temple right there, it would hurt, and it would hurt immediately, right? And that's because the sensation of that is kind of guided by myelinated neurons. So it's a very quick, very insulated response. It gets to the brain very quickly. But after I thump you, if I thump you hard enough, a couple of minutes later, it's still going to be kind of sore, right? It's still going to be kind of a throbbing pain. That is where the C fibers come into play. So if I hit that tissue hard enough, that tissue could be damaged. And because it could be damaged, it is going to, that information is going to travel up to the brain through these C fibers, and they are unmyelinated. Why? Because they're not really in a hurry, right? Because they're just kind of communicating that this part of our body could still be damaged. It could still be vulnerable, and that we need to favor it. We need to be careful. So, for example, if you, and I know this might be a little bit of a gross situation, but let's say you stub your toe really, really hard, right? If you stub your toe really, really hard, it'll hurt for a couple hours. And the reason why is because if you stub your toe hard enough, it could be broken, right? And if it could be broken, you don't want to stub it again. Because if you stub it again and you keep stubbing it, when it's broken, you might lose that toe, right? And so basically the C fibers are going to make sure that we know that this part of our body we need to be protective of. So think of it as like the A delta fibers lets us know that pain has occurred. The C fibers are going to let us know, oh, okay, we might need to play safe with this particular part of our body. So those are the two different kinds of pain receptors. Now, there are other parts that contribute to pain. So part of it is what we just mentioned, how the body is reacting to potential tissue damage. The other part of it is kind of this cognitive emotional response, where we have an emotional response to this pain. So if you're really, really scared and you have some kind of tissue damage, it's going to feel like it hurts more than if you're not scared at all, right? It's kind of a mind over matter situation. I don't want to call it mind over matter because I feel like that usually has some kind of mystical, almost spiritual kind of connotation, but that's not really what it is. Basically, and we'll talk about it in just a moment, but basically your cognitive network can choose to allocate attention away from those vulnerable spots, or those spots that are hurt or are about to be hurt, and because of that we seem to detect it a little bit less. So that is an emotional response that we have to pain, which we'll get into a little more detail in just a moment. So there are different areas of the brain that are responsible for pain. So it's not just the S1 and not just the S2, so the parts of the somatosensory cortex. There are other areas as well, and you can kind of see some of those here in this layout. So this was from a review paper that kind of looked at lots of different studies that have people experiencing pain and seeing what parts of the brain are active, and you can kind of see there's lots of different parts of the brain that are responding to pain. And so just kind of, the ones that I think are some of the most important, we have the anterior cingulate, which you have right here, the ACC. And this is an important spot, and I should reiterate, right now I'm talking not about just the touch sensation, which is what's going on in the somatosensory cortex up here, but just the cognitive aspects of it, so that quote-unquote mind over matter aspect of it. So the anterior cingulate, this is going to be to help kind of change what you're paying attention to. So It is an area that is very closely connected with things like the hypothalamus and the amygdala, which are going to be used in emotion regulation. And so the ACC kind of helps direct our attention to these. It helps us perceive what might be considered unpleasant. If you know anything about the amygdala is really key in detecting unpleasantness, anger, scary things like that. And that has a very close connection with the ACC, the anterior cingulate cortex. The other really important part is the prefrontal cortex. And the prefrontal cortex is this area that you're going to see right up here, right in the very front of your brain, that is responsible for cognitive control. So if you decide that you are going to focus on something else, that you're going to concentrate on something else, that can dull that pain, but can also work in the opposite, where if you focus completely on it, it actually may make that pain worse. So for example, if you stub your toe and you're thinking about your toe and how it might be broken and how it feels, it's going to feel worse than if you were to focus on something else entirely. So that is some of the cognitive aspects of this. One kind of fun fact about this is that when people started first discovering this, kind of that there are other areas of the brain that kind of help mediate pain, like the ACC, the insula, the prefrontal cortex, and the amygdala, they called it the pain matrix. So a matrix being like kind of a, lots of different things combined into something new. And of course, when people responded to that paper, they would always title their journal articles The Pain Matrix Reloaded, because scientists are really dorky and like really bad puns. Alright, so, and yeah, when's the last time you thought about that movie, right? Matrix Reloaded. Alright, so let's talk a little bit about pain sensitivity. So, when we're talking about pain, that kind of tissue damage, that is nociceptive pain. So, when we know that there is some kind of pain that's happening right now, that's nociceptive pain. But after that happens, sometimes we can become more or less sensitive to that area. When we become more sensitive to it, that's called hyperalgesia. And so, again, thinking about your big toe, you stub it and it feels very sensitive. That's because basically your body is telling you, be careful with this particular set of tissue. Is vulnerable right now. But we also have a brand of pain that doesn't really have an external component to it. And that's called neuropathic pain. And this is much more complicated because neuropathic pain is much harder to diagnose. And so when you hear people talk about fibromyalgia, or they talk about chronic pain, or they talk about chronic tiredness and things like that, those are much harder to diagnose because you can't directly see them with your eyes. They are a form of pain that don't have an external source. So it could be that part of your nervous system is damaged in some way, or that it is firing the wrong signals, and that registers as pain. And so because we have these different kinds of pain, where we have on one hand this sharp immediate pain, the nociceptive pain, we have the kind of longer lasting soreness and sensitivity, the hyperalgesia, and the neuropathic pain. There's not one pain medication that's going to work for all different variations of pain. It's really, you need different medications for different sources of pain. Alright, so that's pain sensitivity. One thing that I wanted to mention was that there are now lots of good evidence for not just, and I talked about this in the first video, not just somatosensory touch, so not just the Merkel cells and all those mechanoreceptors, thermal cells that detect hot and cold and pain. There's also cells that detect pleasurable experiences. And so this is kind of surprising because we didn't always think that this really existed. We thought there were only a set finite number of ways that we could be touched or experience touch. But there are certain unmyelinated fibers that are responsible for responding to very slow, very light touches. So you can kind of think of it as like, if you have your arm out and you just very, briefly kind of glance over it like that, it feels kind of good, right? It tingles a little bit. And same with like your face. If you just kind of go like that with your face a little bit, it kind of has that tingly feeling. That's pleasure. That is, that kind of funny, good feeling, right? And so those cells respond to that kind of sensation. And the fascinating thing about this is that those cells are not actually being processed in the S1 or the S2. They're actually being processed in the orbital frontal cortex, which is much closer to the prefrontal cortex up in the front of the brain. So handled by a different part of the brain altogether. I wish I could tell you more, but right now we're just really discovering this kind of cell, so there's a lot that we don't know about it. And the final thing that I want to talk about is sensitivity. Sensitivity and how we kind of experience the what and the where of different kinds of touch sensations. So and this goes back to things that we've talked about in the first and second videos, that we are sensitive in different areas of our body, right? So if you think about it, what areas of your body do you think are more sensitive? So if you think, like, you close your eyes and somebody takes a feather and they're going to touch you in different places, which ones do you think that you would most accurately know when you're being touched? Probably on the eyelid, on the lips, maybe on the neck a little bit, on the earlobe, on your fingertips. And those are the kind of the big ones, right? If you're being touched on your knee, on your elbow, on the back of your shoulder, you're not going to be as sensitive there, right? And so that is partly a function of how closely together those cells are. So, like, for example, the Merkel cells and the Meissner corpuscles, they're just closer together because they have those small receptive fields. The higher the concentration, the better you are, or the more sensitive you are in that particular area, and correspondingly the more parts of your brain, or the more surface area of the brain, is going to be covering that in your somatosensory cortex. And so when we're measuring kind of how much space can you detect differences in on your body, people or scientists use the 2-point threshold. And 2-point threshold, just in case you don't remember because we talked about it in chapter 1, is whenever you take, like, for example, 2 little pens, or maybe that sounds bad. You take 2 little, 2 little sticks, I guess. You take 2 little sticks and you put them somewhere on the body. And the question is this one stimulus or is it 2 stimuli? If you have these 2 sticks and they're very close to one another, you'll just detect it as one. And if you don't believe me, take your 2 fingers and just put it right here. It feels like one thing, right? It just feels like one thing, but you can kind of go and touch your fingertip, and then you can feel, oh, these are actually 2 fingers that I am being touched with. So that's the 2-point threshold. And so right here, it only feels like one stimulus. But if I were to move my fingers a little bit, now it feels like 2 stimuli. Now I can detect that there are 2 separate, different stimuli. So the minimum distance in which I can detect that there are 2 instead of 1, that's the 2-point threshold. And just like for pressure, it varies all over the body. And so you can kind of see an illustration of what I was just mentioning right here, where we have very closely compacted cells in our fingertips. So we have a very low threshold, so we're more sensitive, versus on our wrist where we have a very high threshold, where we are less sensitive. And you can kind of see here on this graph where we have the different thresholds, and you can see that the lowest thresholds means that we're the most sensitive. And where do those occur? Well, the fingers, the thumb, the cheek, the nose, the upper lip, and part of your toes, right? The ones that are least sensitive, the shoulder, the breast, the back, the thigh, and the calf. We can distinguish when something touches us within about a 5 millisecond difference. And that contrasts with vision and with hearing being a little bit faster than that, vision being a little bit slower than that. But over time, we will actually lose some of our sensitivity to touch. And this is especially bad for people who later in life lose their sight. Because of neuroplasticity, our brains can rewire themselves, right? Awesome. But that starts to slow down as we get older. And so for people that lose their vision late in life, usually they might be less able to read Braille, for example, because they just have lost the sensitivity in their fingertips. It's not expressed in their brain, and because of that, they're not able to detect those tiny differences between those bumps there. And so that is like one of the tragic things about losing sight, is that if you lose it early enough, you can become very adept at reading Braille. Later in life, it can become impossible. And finally, the what versus where pathways. There's not a whole lot to talk about here, to be honest with you. The book does talk about some of these things, but I don't want you to have to focus too much on it, except for the things that I'm going to cover in these slides. Because when we talk about where something is, well, that's just kind of, where is it in our body, right? So, like, for example, when we are talking about hearing requires us to kind of use two ears, right? Or if we're talking about vision, it requires us to know, to have two eyes, to know where something is in our visual field or around us in our auditory field. But for touch, well, we know that something is on our forearm because it is stimulating that part of our forearm, and that has a direct connection to a part of our brain. Okay, great. But the question of what something is a little bit more complicated, because there are some things that will pop out to us. So, for example, we're really good at recognizing the geometric properties of something through vision. Much harder when we are just using touch. So, for example, if I gave you, let's say that I gave you, I don't know, just a random toy. If I gave you a random toy, like this, for example, but I'd never let you saw this, I just gave it to you, and I said, all right, what is this? You would feel it, and you probably come to a pretty close, guess that this is a Rubik's Cube or a puzzle box or whatever. But most things are not as immediately identifiable as this, right? If I gave you a cup, for example, you could look at a cup and immediately tell me what it is. With touch, you'd have to kind of move around it, feel on it a little bit to come to your conclusion. But with touch, there are some things that we are better at. We're better at detecting differences in texture and in surfaces with touch. So we can detect very quickly whether or not something is rough versus smooth with touch, whereas it's much harder with vision. We can tell if something is hard or soft, or cool or warm with touch, when that's much harder with vision. So here's a kind of cool example that the textbook talks about. If you were to close your eyes and to rub against any of these stencils, it's really, really hard to figure out what they are. Almost impossible. But if you were to look at them, you can easily tell me what each of these things are with no problem whatsoever. And so there are parts of our brain that help us identify this information. And so just like with vision, we can actually lose the ability to do this in what's called tactile agnosia. And tactile agnosia is whenever we have an inability to recognize things just by touching them. And it is hemisphere specific. So for example, if I had a certain brain lesion in my left hemisphere, I'd be unable to detect what something is with my right hand. But if I had my left hand, I'd be able to tell because my left hand goes up to my right hemisphere. So I would be able to recognize it visually, just not really tactily. And so there's a cool case study that the book talks a little bit about. You can kind of see more of that here, where this individual, where they're touching this and they're asked, all right, we want you to touch this and then to draw what it is that you're touching. And so here, this individual had brain damage in their left hemisphere. So I think it's a he in this case. He's touching this kind of thing with his right hand, and wait, I'm sorry, I got it backwards. This individual has damage in his right hemisphere. I don't know if I said that earlier. In his right hemisphere that is damaged. So he's feeling with his right hand, and because his left hemisphere is intact, he's able to draw it no problem. But whenever he's touching it with the left hand, which is supposed to be processed with his right hemisphere, because it's damaged, you can see it doesn't really look anything like what it should, right? And the same for this piece right here. With his right hand, he can very clearly draw what it is. With his left hand, not so much. All right, so that's all that I want to cover for this chapter. Next chapter, we're going to be jumping into olfaction, which is going to be our first chemical sense. So that's very exciting. So I will see you there. If you have any questions, let me know. Bye.